very much for the invite to attend here. So I joined Fight for Sight a couple of months ago. So this is my first meeting actually at a patient group. So I'm really pleased to be here and actually hear firsthand from patients of their experiences. So I'll just talk you through a little bit of the research that's currently happening in keratoconus and some of the work that we funded previously. So really, how does Fight for Sight support keratoconus research? So we have a number of funding schemes that are open to researchers, as Michelle said, from academics, clinicians, based in universities and hospitals throughout the UK. We have project grants, which are 3 years, which provide funding for 3 years. We also have PhD studentship schemes, and also a small grant award scheme, which is for a 1-year award. And actually, this is the first year, as Michelle and David had said, that we're partnering with the keratoconus group. So, you know, with your generous support, we will be able to make an award this year, and we can't make the announcement quite yet. It still needs that final approval. So you'll find out as soon as we do, but there's definitely one project that will be funded, and we'll let you know as soon as we can. So thank you very much for all the fundraising and hard work that goes into giving us the money to fund this award. So some of the things that we funded in the past include this PhD studentship that went to Professor Inglehern, and it was trying to look at the genetic basis for keratoconus and the implications that this might have for treatment. So as Michelle said, you know, gene therapy and genetics is a really ongoing and really trendy area at the moment, because people see the potential for this having a really big impact and the types of treatment that we can offer to patients, particularly in a personalised way. If we can understand the genetics that underlie a disease, hopefully we can really provide targeted treatment for patients. So this project, sorry, I'm stuck in on the suite that I was handed earlier. This project looked at the underlying genetic factors potentially in inherited eye diseases passed down through families. And the PhD student looked at samples from 15 families, and they discovered 2 new genes that people hadn't realised were implicated with keratoconus before. But it's a very rare cause of the condition. And I think what this does, along with other studies that have happened in this area, is really show that keratoconus is a very complex disease. There are lots of genes involved, but it's not clear that any one gene has a greater impact than the other. But it's still very useful information, and actually there's a lot of ongoing studies now that are doing larger studies with whole genome sequencing and very complex, kind of trying to understand big networks and so on, to try and understand if there are other genes that we can target for treatment. Another award that we made was to Mr. Daniel Gore. And so, as we know, the collagen cross-linking has really taken over, I think, in the past few years as a real game changer in keratoconus. So 9 out of 10 cases, it does stop disease progression, but there is, as with any treatment, a risk of complications such as infection or scarring. So this study was trying to look to see if there was a more safer and less invasive way of actually applying the vitamin B12 to the cornea rather than through cutting the epithelium, which is where you can get some of the infections. So they looked to see, so they used a very trendy kind of microscopy technique to see how much vitamin B12 actually gets to the cornea via just dropping it on with drops compared to actually putting it on underneath the epithelium. And this found that actually it was much more effective to cut the epithelium and then apply the B12 that way, vitamin B2 that way. But actually what's quite nice is a further study that we funded later on from Professor David O'Brat, actually found a different way of applying riboflavin to the epithelium. So in this one, actually the cornea and the eye, it has an electric charge going through it that we aren't aware of. And so they try to alter the way that the vitamin is applied, and they use a technique called iontophoresis, which kind of puts a charge through the vitamins, and then they found actually by doing that they were actually able to reach the cornea without having to cut through it. And what's nice is that the results from this initial study show that epithelium on, kind of cross-linking as they call it, is just as effective as the epithelium off technique. So now they're trying to make this into a larger scale clinical trial. So hopefully by the end of this process we'll come up with a way of doing collagen cross-linking that isn't as invasive and doesn't require surgically cutting the epithelium. And doing something different, actually, is a PhD studentship that we then later funded to Professor Daniels, which I think a lot of people have heard about stem cells and, new kind of techniques and therapies around that. But, so stem cells are really a type of cell that has the potential to differentiate and become any other kind of cell really in the body. And the eye has its own supply of stem cells. It has stem cells that are in the cornea and then some that are in the epithelium. And what they were trying to do in this PhD studentship was find out the best way of being able to extract those stem cells from the eye, then to grow them up in the laboratory, and then put them back into patients' eyes to hopefully then cause a regeneration and a self-renewal of the cells that are missing. And by the end of this PhD studentship, so the student only finished last year, and she was able to identify the best location for getting out and obtaining the stem cells. And they're actually now developing this into a protocol for a stem cell-based clinical trial, which they're hoping will be available in 2 years' time at very early stages. So this is really exciting, and it shows the way that even students and PhD students, if we apply some focused funding, they're really able to achieve a lot. So, moving on to clinical trials, we recognise that we as Fight for Sight can't do everything by ourselves, and there's a lot of research that's happening. We do fund everything across the scientific pipeline, as we call it. So, basic research trying to understand underlying mechanisms and, the genetics and the stem cell therapies, and all the way through to the translational and actually to the clinical studies, and where it has impact on patient benefits. So clinical trials are really the research studies involving patients, which compare a new or different type of treatment or therapy against what's the standard of care, essentially the treatment that patients are currently receiving. And all new treatments or interventions have to go through a clinical trial process for regulatory reasons and to make sure that they are actually better or more effective and safe than the current treatments out there. And what's quite, and I did a quick search before I came to this meeting, and keratoconus is actually an active area of clinical studies. There are, from the government perspective, the NIHR, which is the National Institute of Health Research, they're really the organisation tasked with the delivery of research within the NHS. So any clinical trials that are conducted in an NHS hospital have to be registered with the NIHR, and they run a website where they list all the clinical trials that are running in the UK. So there's 43 studies that are currently listed on the NIHR website that are relevant to keratoconus patients. And internationally, actually, there are over 160 trials. So, 41 of these, of the 160 trials, they're either recruiting at the moment or not yet recruiting. And by not yet recruiting, we mean these are studies that have been approved for funding, but they're not ready to start up yet. So they're looking to recruit patients very soon in future. And in the UK, there's 6 trials that are currently recruiting keratoconus patients. If you're interested in finding out more about the clinical trials that are running in the UK, this is the website, this is where you need to go to. It's the UK Clinical Trials Gateway, which is run by the NIHR. And if you go there, you'll be able to see the different trials that are running for a particular condition. What's important to note, however, is that it's really important that you speak to your clinician. You can see what's out there, but your clinician is really the best person that you need to speak to find out what might be suitable for you, because not all trials will be open in all locations or be suitable for all patients. So some of the, so I thought I'd pick out a couple of the current clinical trials in the UK. So the group from Liverpool today, if you were in the other room earlier, were talking about an OCT technique to try and detect keratoconus earlier. This is another trial which is looking to use kind of ultrasound to try and detect keratoconus earlier. And interestingly, this is actually an international study. So when we talk about studies, I think one of the really good things about research actually is that it's a collaborative nature. Often we know there's some things that we can't achieve by ourselves, and we need to work with our partners both in Europe and internationally. And this study was actually set up by a US group. And they then opened it up to a UK institution as well to allow patients to be recruited into this study in the UK, so that jointly between the US and the UK, they can recruit the number of patients that they need to achieve the outcomes. So they're aiming to recruit 250 patients internationally, and they're recruiting till the end of September 2019. And the investigators here have developed a technique using ultrasound for imaging the cornea and measuring the thickness of the different layers, including the epithelium and the stroma. And the idea is hopefully to develop a technique for early diagnosis, which would then, of course, allow for earlier treatment and intervention. Another one, I won't say the name fully, so it's a study that actually was run by Professor David O'Brott, who we have funded as well, and it's currently open. It's aiming to recruit 36 patients, and it says that the recruitment is ending in September, at the end of this month, but actually we know that it has been extended slightly because they need to recruit a few more patients. But in this, really, they're looking at standard collagen cross-linking. The UV is applied directly just onto the middle of the cornea. But actually what they're saying is if you apply it in a more targeted way in the area where the cone is or where the bulge is, whether that's a more effective treatment than just giving it kind of in a general way. So they're trying to determine the location of the cone based on tomography to get a 3D image, and then apply the UV in a much more targeted way. So, other keratoconus research. So again, I had a quick look, and there are over 6,500 papers that have been published in keratoconus. So this is a really active field of research. So 367 of those papers were published in just 2018 alone. So in the past 9 months, there's been nearly 400 papers published in this area. A lot of it is clinical studies. A lot of, I suppose, emphasis have been put on management and treatment of keratoconus, and obviously we've seen a lot about collagen cross-linking recently. So a lot of the papers are around, is this method of collagen cross-linking better than the other? But and there's a fewer studies around understanding the causes and mechanisms of disease. But what has been good is there's been a recent focus on genetics, and there has been a real push to try and understand some of the underlying mechanisms. So one interesting study, which actually was a systematic review. So basically, they pulled together the published data from a load of different papers. So in this case, they reviewed the results from over 340 keratoconus related articles. And they tried to map to see, are there any genes that they can work out were correlated based on the different published results. And they found actually that keratoconus can be related to some other diseases, and it shares some of the genetic traits as other diseases as well. But it did allow them to pull out, some genes that might be a target for future research, really. Looking more at kind of collagen cross-linking, I think the focus has been a lot on the clinical application of collagen cross-linking. And one interesting paper that I found was actually trying to look at the underlying mechanisms as to how it works. Often, with treatments, we provide the treatment, but we don't fully understand exactly why it works or how it works. So this was quite an interesting study that because they were trying to understand how collagen cross-linking works in order to improve the treatment and to stop some of the side effects that you might see. So they found that actually collagen cross-linking is really effective at organising the collagen structures within the eye, but also that it was really good at protecting the extracellular matrix, which is basically the environment in which the cells sit, and it stops the extracellular matrix from degrading. And so by stabilising that area as well as stabilising the cells themselves, they think that's how it works. So this is an interesting area because it means that we'll be able to look to see if there are other proteins that are involved in collagen cross-linking that could be, really utilised to make the treatment better. So I think my time is up, and I'll leave it there. But I just want to, this was a whistle-stop tour through the research. So, just to really leave you with the message that, keratoconus is really an active field of research for science and for Fight for Sight as well. And actually, by coming here, it has been great for both Michelle and I to understand some of the challenges that the community is facing. And we're really keen, I think, we want to develop our patient engagement activities. So if any of you are interested in working, with us, we'd be very pleased to hear from you as well. So thank you very much.